Here is the counterintuitive takeaway. DNA computing is real, but its best near-term use is not fast thinking. It is ultra-dense memory and chemistry-native logic wrapped in a very hybrid system. As of 2024, the field had enough progress for a 10-year systems-level review of DNA data storage. That matters because it signals a shift in mindset. The hard part is no longer proving DNA can hold bits. The hard part is building an end-to-end machine that can reliably write, store, and read information at scale. Start with what makes DNA attractive. Material density is the headline. Nucleic acids can pack information into tiny physical volume. They also offer massive chemical parallelism, meaning many molecular events can occur at once. In principle, that combination changes what storage and computation can look like. Now comes the reality check. Molecular systems are constrained by reaction kinetics and diffusion. They are also constrained by stochastic error. Molecules degrade, samples get contaminated, and the interface between molecules and electronic control systems is slow. So the most important insight for strategy is this. DNA-based information systems are not a device, they are a workflow. The bottlenecks live in encoding, synthesis, storage stability, targeted retrieval, sequencing readout, and end-to-end error correction. If you only optimize one step, the system still fails. That systems framing also explains why the practical stack remains hybrid. Conventional software designs sequences and protocols. Laboratory automation or manual methods handle synthesis, extraction, amplification, and sequencing. Then electronic compute decodes, verifies, and reconstructs the result. The molecules do not replace the data center. They become a new physical medium inside a larger pipeline. Once you accept that, the next question becomes, which workloads actually belong in chemistry? DNA computation is most credible where the work is chemistry native. Think molecular verification, rule checking, analog signal processing through concentrations, binding affinity computation, and structured biochemical reaction systems. In these cases, you are not translating a fast electronic algorithm into wet lab theater. You are letting molecules compute in their native language. There is a proven example of this bounded approach. In 2008, DNA computation was applied to molecular verification of rule-based systems. That is a very specific kind of workload. It is symbolic verification, but implemented through molecular operations in a wet lab paradigm. The lesson is not that DNA will run your general software stack. The lesson is that certain verification tasks can be mapped into molecular procedures when the structure of the problem fits. A second proven category is analog molecular computing. DNA-based analog computing methods were formalized, emphasizing concentrations, reactions, and molecular dynamics as computational variables. That is a different mental model from binary gates. You are not asking for clock speed. You are asking for physical dynamics that naturally encode a computation, often as part of an assay. A third area is DNA as a programmable material. Methods for synthesizing long DNA-based nanowires were published in 2011, with additional nanowire synthesis methods published in 2018. These results show nucleic acids can serve as engineered structural or templating elements. That is valuable for nanoscale fabrication and sensing concepts, but it is not yet a high-speed electronic interconnect substrate. Strategy teams should treat DNA wiring as specialized structure, not as a rival to semiconductor wiring. If you want the clearest picture of near-term limits, ignore the glossy diagrams and look at operations. Reagent cost matters, sample preparation matters, extraction yield matters, contamination control matters, read and write latency matters, molecular degradation matters. On top of that, there's computational overhead, because you must detect, classify, and correct biochemical errors. Notice what that implies for procurement and vendor claims. You cannot price DNA storage as cost per byte in isolation. The relevant unit is the full write, store, read loop, including synthesis, extraction, sequencing, error correction, sample handling, quality assurance, and data reconstruction. The question is not whether a lab demo wrote data into DNA. The question is whether an institution can retrieve it reliably, years later, under real governance constraints. Error correction deserves special emphasis, because the biochemical channel is messy in a distinct way. You can see insertions, deletions, and substitutions. You can see degradation. You can see contamination. You can see sampling failures, where the molecule you need is simply not in the portion you retrieved. Those error modes interact across the whole life cycle, not just at readout. So the architecture of error correction must match the full storage cycle, not a single step. Now widen the lens to the ecosystem that shapes what is possible. DNA storage and molecular compute depend heavily on the read path, meaning sequencing instrumentation and its economics. They also depend on the write path, meaning synthetic DNA manufacturing capacity. Between those endpoints sits the messy middle, the lab infrastructure layer, including enzymes, reagents, polymerases, and sample prep instruments that determine whether workflows are reproducible. That is why the leading actors look less like chip companies and more like a hybrid of compute research, DNA synthesis, sequencing platforms, and lab infrastructure providers. Microsoft Research has worked on systems integration and coding theory for archival storage. Catalog has pursued commercial DNA-based information storage workflows. Twist Bioscience is relevant to synthetic DNA manufacturing. Illumina shapes much of sequencing's throughput and cost structure. Thermo Fisher Scientific anchors a broad reagents and instrumentation layer. Oxford Nanopore Technologies adds a different sequencing platform, including portable sequencing. Integrated DNA Technologies supplies oligonucleotide synthesis and molecular biology reagents. Public institutions also matter, especially for standards metrology and scale-up capabilities, including NIST, U.S. Department of Energy National Laboratories, and DARPA. That mix leads to a governance point that many leaders underestimate. Molecular information platforms are dual-use by default. When you combine synthetic DNA, automated wet labs, and high-throughput sequencing, you are operating near the same capabilities used in advanced biotechnology. So the governance model cannot be an afterthought. Without careful policy, every storage or compute workflow risks becoming a biosecurity exception process, which can stall adoption and create brittle compliance regimes. Standards are the quiet foundation here. Cross-laboratory reproducibility requires reference materials, encoding formats, error reporting, contamination controls, and life cycle metadata. Without those, results cannot be compared, audits cannot be performed, and long-term archives cannot be trusted. For an investor or sovereign strategist, this is a familiar pattern. The biggest leverage is often in measurement, interfaces, and accountability, not in the flashiest prototype. So what does the time horizon actually look like? From 2026 to 2050, DNA-based storage can become a specialized archival tier for high-value, cold, long-retention data if cost, automation, retrieval selectivity, and error accounting improve enough to justify lifecycle integration. That is a very specific promise. It does not mean you replace cloud storage. It means you add an archival layer for data worth preserving but rarely accessed. From 2050 to 2100, hybrid molecular electronic systems may handle selected chemical, biological, storage, sensing, and verification workloads. The keyword is selected. Molecular parallelism or physical density must offset latency, and systems must be robust enough to operate outside bespoke lab conditions. Beyond that, the long horizon gets stranger and more speculative. Molecular compute could be embedded in synthetic cells, living materials, environmental sensors, and slow autonomous biological infrastructure. Yet even then, it is not positioned as a replacement for high-speed electronic or photonic reasoning, and general-purpose DNA computers replacing CMOS at production scale remain speculative. The evidence supports bounded molecular computing, not universal high-speed molecular processors. For capital allocation, the practical guidance is straightforward. Fund molecular information systems as archival storage, wet lab automation, biochemical sensing, and programmable life infrastructure. Do not budget them as near-term replacements for GPUs, CPUs, photonic accelerators, or industrial inference factories. For operators, the sharper questions are also simple but uncomfortable. Which data has retention value high enough, access frequency low enough, and governance maturity strong enough to justify a DNA storage pilot? Does your organization measure reliability across the full loop? Write, store, retrieve, read, decode, and verify, or only at isolated steps? Which workloads are genuinely molecular in structure, and which are electronic workloads being inefficiently translated into chemistry? And finally, who owns failure accountability if a molecular archive is unreadable decades later? The memorable line is this: molecular compute will not win by being faster than electronics. It will win by being denser than matter and more native to chemistry than code.